Hi, we're here today talking about viruses. This is just a quick overview of some of the key points of viruses, their life cycle, and the differences between the different types. So if we start off and just take a look over here at our picture, the virus you see here is a bacteriophage. And these bacteriophage, which kind of look like little lunar landers, seem to all have been infecting this bacteria because viruses are very specific. They're specific in the host that they do infect. Okay? And bacteriophages infect bacteria. And while viruses have kind of a really bad name, the truth is that bacteriophages are a really important tool in biotechnology, in gene cloning, and those sorts of things. So they do have applications that have been of tremendous benefit to humankind. But here you can see here these viroids all attaching to this bacteria. Now, viruses, hotly debated in the scientific community, is the question, are they alive? And we know that in order for something to be alive, it has to meet all of the characteristics of life. Viruses are unable to reproduce on their own. They have to infect a host cell in order to reproduce. And this is one of the reasons that they're not considered living things. They're considered acellular. And let's talk a little bit about their composition and some of the different types of viruses. So viruses come in all kinds of shapes and sizes, different types. And you can see a few listed here. The tobacco mosaic virus, adenoviruses, the influenza virus, and bacteriophages. Again, they're all very host specific, but they do have a few things in common. Number one, viruses are surrounded by a capsid. And this capsid is the outer covering of these viruses, and the capsid is made of viral proteins. Now, some viruses also have this membrane envelope, and you can see that here in the influenza virus that surrounds the capsid, but that's not found in all viruses. So we have the capsid. And then we have the genetic material. And the genetic material can either be DNA, or in the case of retroviruses, it can be RNA. Retro because in order for that virus to start to manufacture more viruses, the viral RNA is actually converted by reverse transcriptase into DNA, and then that DNA codes for the new viral proteins. AIDS, the HIV virus, is a great example of a retrovirus, and we'll talk a little bit about that in a minute. Now, if we take a look and just focus over here at our bacteriophage for a moment, the bacteriophages almost work like injecting through a hypodermic needle. The bacteriophage has these tail fibers, and you can see them here in both the electron image and the diagram here, and these tail fibers attach to the cell membrane of a cell. Okay. And then the viral DNA, which you see here in the capsid, is injected down into the host cell and then can begin its life cycle inside that host cell. Other kinds of viruses, like the HIV virus, actually dock with some surface proteins on the cell that they're infecting. And then once they're recognized, are incorporated through endocytosis right into the host cell. So a couple different mechanisms of insertion there. Now, viruses have two different styles to their life cycle when we're talking about these phages. They can either be a lytic lifestyle or lysogenic. Two different strategies here. So here we have the phage, which is docking with its host cell, and you can see here that that viral genetic material has been injected into the cell. And you can see that here, and it's shown in blue so that we can follow it throughout the whole process here. Once that's injected in, we have two choices. We can either go with the lysogenic style or we can go with the lytic style here. Okay. In the lysogenic style, what's going to happen is that that piece of viral DNA is going to get inserted into the host cell's genome. So you can actually see here that it's spliced and inserted right into the host cell's genome. And it can lie dormant there for a number of years. This is actually what establishes lifelong infection. The idea that that viral genetic information actually inserts itself right into your very own DNA. Because the virus isn't actually being expressed at that point, your immune system is unaware of it. It's not attacking and destroying those cells. It's like a sleeper agent. It's hiding in those cells. And every time that cell replicates, we can see here an example of binary fission. As that cell divides, we know that the first step is that it's got to replicate its DNA. And as it replicates its DNA, look at this, it's also replicating the viral DNA. So now we've doubled it. So now as it splits into two new cells, we have two cells, and this cell is going to have be infected with the viral DNA as well. And we know that bacteria reproduce very quickly, and so as they continue to reproduce, we're now spreading this viral DNA throughout, and you can see that here. Now, for a variety of reasons, and it's still an area of active research, but there's a variety of conditions which can cause this DNA to now be expressed. So if the DNA starts to be expressed, we leave the lysogenic lifestyle and we go to the lictic lifestyle. Lictic meaning that now the viral DNA is going to start to be expressed. Okay? So the viral DNA is excised out often of the bacterial chromosome. We start to manufacture the viral proteins for the capsid and the viral DNA. We then assemble the phages, and these eventually cause the cell to lyse. The cell bursts and the phages fully formed then emerge from this. So a lictic lifestyle causes the host cell to lyse and the viruses emerge from that. The lysogenic lifestyle is when the viral chromosomes, the viral DNA, is incorporated into the host cell DNA and then is multiplied and multiplied and multiplied as that host cell goes through binary fission or mitosis. So let's take a look at a variation on this. The HIV virus, human immunodeficiency virus, is a retrovirus, meaning that its genetic info is RNA instead of DNA. Okay. Like other viruses, we have a capsid. We also have this viral envelope around the HIV virus. What happens is that there are two cell surface receptors that are found on your cells. When the HIV docks with the white blood cells that have those two receptors, its protein receptors recognize those two. You need to have both of the receptors in order for HIV to infect a cell. When they're both recognized, the HIV virus actually merges, and its membrane merges with the membrane of the cell membrane, the host cell, and the interior portion of the virus is actually inserted into the cell. You can see here that what's coming in is the capsid and the viral RNA. It also brings in it, in HIV's case, reverse transcriptase molecules. Now we know transcription takes DNA and makes it into RNA. Reverse transcriptase takes RNA and manufactures DNA from it. So in comes reverse transcriptase. Using the machinery and using the nucleotides found already in the host cell, we manufacture this RNA-DNA hybrid. Okay. We then use that DNA strand to manufacture a second strand of DNA, and it gets inserted right into the chromosomal DNA of the host cell. Okay, it gets inserted right in there. Then, now we've hijacked the cell's machinery. So now the cell is triggered into starting to manufacture the viral proteins that are going to go on and be the capsid. to manufacturing reverse transcriptase, and to manufacturing the various proteins that are going to go into the viral envelope and the RNA as well. In doing that, these then assemble inside the cell, and you can see coming off the bottom here, these bud out of the cell via exocytosis. So the cell isn't actually lysing here, it's not bursting, but we've totally hijacked the cell's machinery here and are tricking it to produce the HIV virus, which is then being pumped out of this host cell. And here you can see the electron micrograph images of this. So here we have HIV docking into that white blood cell. Coming into the cell, we have the capsid and the RNA that it encloses, okay? And then those newly formed HIV viruses are going to now bud off of the cell via exocytosis. Now, Howard Hughes Medical Institute has a really great video on this. They have a ton of great stuff. I highly recommend that you check them out, HHMI. But I thought that I would like to show you this video clip now.
So this is HIV. It's a typical retrovirus, meaning that it has an outer envelope, and in the center it has 2 copies of RNA, as well as an enzyme here in blue that's reverse transcriptase, which will ultimately turn that RNA into DNA. The virus itself, with this outer envelope protein, actually directly infects T helper cells. The way that it does this is that as it comes up to the cell surface, it uses receptors that are on T helper cells and exclusive to T helper cells, which are CD4 molecule, which really defines T helper cells. It's a surface receptor that binds to the envelope protein. That causes a conformational change and allows a second receptor to grab hold of the envelope. This is the chemokine co-receptor. It's also called CCR5, and we'll talk about that more. What happens now is that the stalk of the envelope protein pierces through from the virus into the host cell and starts to draw the cell membrane and the viral membrane together. And what ultimately happens is fusion of those 2 membranes, and the viral genetic material is injected essentially into the cell, and the envelope protein is left at the cell surface. The virus has a matrix and a capsid protein, shown here in green and red, that essentially are digested when it enters into the cell. That releases viral enzymes and the viral RNA. And here we have reverse transcriptase, which takes the viral RNA and, using host nucleotides, converts that viral RNA into a single strand of DNA. While it does that, it makes some random errors, which is characteristic of reverse transcriptase. It has very poor proofreading activity. That single-stranded DNA now is again reverse transcribed into a double-stranded DNA. At that point, another enzyme that has come in with the virus in the beginning, called integrase, essentially grabs hold of that double-stranded DNA and carries it through a nuclear pore into the nucleus of the cell. Within the nucleus of the cell, it finds the host chromosome, and the integrase enzyme makes a nick in the host DNA and allows for HIV to insert itself into the host chromosome. And that right there is what establishes lifelong infection. Now, RNA polymerase comes along and makes messenger RNA. Those messenger RNAs encode for different viral proteins. They end up associating with ribosomes at the surface of the rough endoplasmic reticulum. And here's a piece of mRNA that's making envelope protein, which is directly produced into the endoplasmic reticulum. And it's shuttled then through the endoplasmic reticulum and taken to the cell surface, where at the cell surface it becomes embedded in the cellular membrane, and at this point, coalescing with other envelope proteins that have been produced, you have this cluster of envelope proteins now on the surface of this infected cell. At the same time, there are other messenger RNAs that are being produced that allow for translation of other viral proteins. So here are additional viral proteins being made, which are going to be used to make up the key components that the virus ultimately is going to need. These are transported again to the cell surface, to the area where these envelope proteins are, and a strand of RNA as well as some of the enzymes are part of that complex. This then buds off at the cell surface at this point, but it's still not a mature virion because the polyprotein chain needs to still be digested into its component parts. That's done by an enzyme called protease. Protease breaks up those polyprotein chains and ultimately allows for them to coalesce and form the mature structures that make up the final virion. And now you have a mature infectious virion that can go on now to infect other cells. Once that happens, now the cell can produce tons of viruses, and this is really what then keeps the whole process going.
I hope you found the video from Howard Hughes Medical Institute informative. Keep asking questions. Check out their site. They've got a lot of really great resources there for you to explore. And enjoy. Thank you. Bye.